is to get a quick summary, a report out of where we're at, not a summary of the discussion necessarily, but in terms of the writing, where are we at in the process, what's needed. I'm going to ask Archie if you would be first to report out on behalf of Precision Management.
We had a really good combined discussion with Precision Selection, and as became clear, I think over the last couple of days, there's a lot of similarity there, building from the same data sources, the same sorts of platforms, the need for the same partnerships in achieving what was envisioned in our discussions for the next 2, 5, and 10 years. So we've decided to combine those sections of precision selection and precision management into one section. I'm confident we can capture the same great points that were made in the management discussion in that combined section, but that's really the primary thing that came out of the last hour.
Thank you. So for genomic and functional biology, Kathy, Joan?
I think we expanded on details and tried to give 2-year and 5-year goals and how we would use FANG data and the importance of things for different species, and the possibility of cells, tissues, whole animal, thinking of things from those perspectives. Anything else, Kathy?
Thank you. Holly, I'm going to head your way next. Or Hans. Okay.
Yeah, so we had a good group, and on the host pathogen interactions, we focused mainly on trying to get more information on rationale as to why we should do this. So we're sort of thinking about fund holders, and we also focused on outcomes that we would have, as well as specific tools to help, guide like the RFAs, et cetera. So we'll continue to improve upon the write-up, and hopefully everybody in the room can contribute. But I think hopefully we've captured most of what we want.
Thank you. Jim, can I come to you for phenotyping?
So for the phenotype discussion that we just got done with Joan, had articulated again, like she had said, 2- and 5-year goals, added on, working towards an outline, if at all possible, between now and PAG, that we've got something for people to continue to build upon. But really building upon all of the discussions we've had for the last 4 days. Yeah.
And I don't see Derek in the room. Okay, we can come back to microbiome, because Tim, you were with Genome Tools.
But I can't talk anymore. The genome tools and resources, we are going to need to rewrite some sections, but we kind of agreed on how to do that, so it shouldn't take me too long. We put down some specific goals that we didn't have time to talk about in the short abbreviated session that we had. That include chromosome level scaffolded genomes for important species, developing new tools for genotyping, new arrays, and things that go beyond SNPs, include other aspects of genome variation, and developing catalogs of extant intra-species variation, DB SNP type things, but moving beyond just SNPs. And our long-term goals of evaluating whether pan-genome methods are of any use, and making sure that people or that species have HAB maps and samples from the HAB map that have known genotypes to evaluate the genotyping platforms that are developed. And our pie in the sky thing was generating synthetic genomes, like we heard about this morning, that allow you to snap things in and out and assess function in that way.
Thank you. Kathy, I'll ask you if you could cover education and training for us.
Well, I don't have a whole lot to add from what we did before, but what we did first, we asked for, Sarah was our scribe, and we asked her for some perspective from a student's point of view, and that was very helpful. And then we kind of came around to start trying to distill our thoughts into some bullet points that we can expand upon. So for the next stage of the writing, again, focused on curriculum, communication, industry partnerships, interdisciplinary training, and those kind of ideas.
Thank you. Okay.
For the characterized animal populations, much like what some of the other groups were talking about, we came up with some actionable bullet points. We're going to continue to refine those and maybe consulting with some of the other groups where we've got some overlap. So.
Thank you. Kurt, we had bioinformatics. Did you want to cover, or I know that you were in the precision selection. He's punting to Jim. Okay. Don't blame him. We had also, while I'm on this side of the room, the genome modification. Kevin, I'm headed your way.
So for the short term, we saw expanding technologies to species that don't have robust technologies as a priority, and also characterization of unintended consequences, whether that's genetic, off-site cleavage, et cetera, or epigenetic associated with the technology overall. I think everyone came to the consensus that we should build either some actual or virtual centers that would provide these services to people at large, people in the community. You want to validate whether or not your causal mutation is actually causal. We can introduce that and provide that service. The bigger picture, though, at the end of the day, is we would really like to find a way to have as policy of USDA to not reject grants out of hand simply because they're genetic modification. And we have no idea how to do that.
Okay. Thank you. And, let's see. Jim, I was looking for you. You're ducking behind.
So for the writing group, some of the shorter term portions that they had was probably standing up workshops for training, both from, well, we talked about it predominantly from more informal, hands-on, hackathon, other actual applications of software, databases, as opposed to the education group that had talked more about, I think, more formalized training in given areas. So this is kind of an interface between the two. Longer term was the need for databases in order to facilitate data reuse, that we have lots of information that is collected for reason X, but it also has a broader context that it could be used in order to answer questions beyond its original portion, and that we've got information that is in a lot of different silos, and we don't have a place that we could physically put it in order for, to some extent, a one-stop shop in order to come find some of these informations. So to some extent, like what Sean was talking about for dbGaP, we don't have those types of resources available for the livestock industry at this point. We really didn't break it out for anything specific for an individual species or anything. We always kept it really quite broad as it was applicable to everything. Longer or shorter term was the need to probably democratize a lot of the softwares in which we have, and that they were built in order to work on an individual configuration for the computer, but they need to be built in such a way that anyone could deploy them on any computer in which they have, and not have to spend time to optimize, reload, and balance things again. There was a need for development of databases to facilitate things that don't even exist today, and the example that was brought up was more the metagenomics, that there's 16S data all over the place, but nobody understands what my OTU1 and your OTU1, are they the same or different? If there was a way in which that data could be brought together and better understood, would be another longer-term goal that would be really quite nice. And the list keeps going on from there, but hopefully we've developed an outline that will get us there in order to articulate it as we go. Okay. What? Oh, for tool reuse? Oh, yeah. So dissemination of the things in which it is that we build, such that we're not just going back and redoing the exact same thing again. Thank you, Kurt. Absolutely. Did that make sense for folks? Yes, no? No. Okay. Right now, how do we take the softwares that Kurt has built a wonderful software to do X, and let's get it out so other people could physically reuse it and build upon and expand upon it to a greater extent? Yeah. Software carpentry to some extent. Yeah.
Okay, Tim, we don't have Derek with us at the moment. Can you recap microbiome for us?
Yeah, I can because it's easy. So that group came to a really nice consensus. We want to do a round of polishing on the document, but we pretty much captured everything. And one of the primary things is that the microbiome work in most livestock environments is still really early, and we want the agency to understand that you need some baseline data before you can really formulate strong hypotheses and test them. So there are a few situations where that's not the case, but generally, broadly, especially across species, we still have some stamp collecting to do before we can launch strong hypothesis-driven research in this area. So we recognize, I think, broadly that it will have importance, but we need to find the support to do that base level, how much variation exists in a normal environment, just changing over time, or in response to normal variation, like it rains in your catfish pond. That changes the microbiome for a while. But if you're sampling longitudinally, that can screw up your analysis if you haven't accommodated for those kind of changes.
All right, thank you. So we've covered a lot of ground. I mean, this really is a big job. I guess now I know exactly why it hasn't been done for 10 years, right? I mean, we've really appreciate all the enthusiasm everyone has brought in the discussions that have gone on. I know we ran on late in the agenda, but I thought it was important to listen to the speakers we have and ask them questions and not want to interrupt some of the discussions. So what I hear from a lot of the groups is that the teams that we're working still have a little bit of work that they want to do in terms of polishing or moving things around before we get to the next step, and so there'll be time to do that. We've been getting some questions about next steps, and tomorrow we'll meet with the steering committee and the writing team leaders that are still in town to talk about next steps forward. You know, clearly we want to be able to take this and to get it in a form that we can present at PAG and share with others in the community, and then have some kind of online platform to continue to get feedback and input. You know, I know that we want to make this a useful document. We want it to be useful for you, for our colleagues around the nation and internationally, but also we want to have something that we can point to let people know what we do and what the capacity is, and frankly what's been accomplished over the last decade. And so you really have given us the foundation to be able to do that. So I'll have a couple more things, but Lakshmi, I wanted to give you an opportunity.
Yeah, I just want to thank a lot of people here. One is goes to Jim Reese, James Coltus, and Nick. Gulherme, so they have put together the grant. It was a very quick turnaround, and I think CARE also was part of it. So I think getting this going, it was very real important key step to get the funding for it to make it possible. So beyond that, I think. I would like to thank the, again, probably I've talked on behalf of both of us, like the steering committee. So the steering committee, and I think, we had, whoever we asked for, like, we picked the best of the minds to be able to, work on these very diverse topics, and we're really very thankful to each one in the steering committee who have agreed to, participate in that. Like, we had very frequent phone calls, and it was a lot of coordination, and I think most of the hard work was done by CARE in getting, doing the work. I think, again, just goes thanks to all the steering committee members. I think a couple of people, especially that are very important and instrumental in getting us organized and having this, like, who did a lot of heavy lifting here, behind the scenes, are, Janice and Lauren Coleman. So again, probably there are other people here also there. Really?
They've really been helping us a lot since July.
Yeah.
When we first started talking about having this activity and include the blueprint activity in addition to 2 workshops in 1 week. So we really do appreciate your effort.
So the other group I would like to thank is our federal partners, FFAR, people from NIH, NSF, DOE, our international colleagues, David Bailey and Jean Charles, who have come all the way, to come and support this effort. I think it has been a big motivation for all of us, and to see that, like, we're just not thinking in a shorter sense, but having a much broader view of the whole federal government and also, international support for what you're doing. Again, a big thank you for all the people who have come in from long distance to be able to participate.
I just have one more. So it was important that we get representation of a lot of groups, and important to the entire steering committee was having some graduate students who could come and participate. But we had to have something for them to do. And so we had some scribes that were with us this week, and we very much appreciate your willingness to come and be with us for a few days and capture everything that came out of our mouths on paper for record, so that we can have this to look back on as we continue to work on the drafts. And so, you've heard a lot of the conversation that we've had about how important it is to train students and have the next generation coming up in animal genetics and genomics. And so that's you guys. So we appreciate your efforts here, and thank you for coming and participating.
Right.
So, let's go to the right. So we had, in addition to the steering team, we wanted to thank our writing team leaders. These are folks that we asked in fairly short order to come up with the drafts. And we knew that it would be important to have something to look at when we got here, something to not start from scratch, but to start with and to be able to improve. And so all the folks that participated in that, we thank you and let you know that you actually signed on for a whole year until this thing's done. So thank you very much. Thank you all for coming. The shuttles will be here around 1:15. So hang out and feel free to